Today we're going to start discussing degradation of the last group of biomolecules who are used for energy production and these proteins. We gave you a review on protein structure, a little bit function last time, so we should all be on the same page. So again, I think I showed this slide one more time when we start talking about electron transport, but as you know, this puts everything in context. Degradation of all the biomolecules that are utilized for energy production, carbohydrates, lipids, and proteins. We can produce acetyl-CoA through their degradation, channel the acetyl-CoA to the citric acid cycle to produce electron carriers that can then be utilized in electron transport and oxidative phosphorylation for the production of more ATP. Okay, so as I said, today we'll talk about protein degradation, and with this, of course, we also talk about degradation of amino acids. However, amino acid degradation or the discussion of the degradation of individual amino acids is rather tiring and let's say boring. Okay. So what I'm going to do is I will just talk about the general principles of the degradation of proteins, and we'll see what happens pretty much to all amino acids as they are broken down, and we will learn a little bit more about the degradation of individual amino acids, most likely on Thursday when Niati, is pronounced correctly, will give a student presentation on genetic disorders of the amino acid metabolism, and there she will give some examples of pathways that lead to degradation of individual specific amino acids. Okay, so if we look at the degradation of amino acids and proteins, we have to be aware of the fact that degradation synthesis of proteins happens all the time in our body. Okay, so there's something that we can call protein turnover, which is constant. So proteins that are required are being synthesized, and those that are not required are broken down. And the amino acid we utilize, either for energy production or for the synthesis of other proteins. When we take our proteins in our diet, then we can utilize the ingested amino acids, obviously, for energy production. To what extent we do that really depends on what kind of organism we are and how rich in proteins our diet is. If, for example, you are a carnivore, some of us might qualify almost for that. We would take up 90% of pretty much our energy requirements from protein. So, typical carnivore, a cat, something like this. Okay, not a house cat necessarily. I talked about energy production in plants briefly before. We learned that lipids, for example, are really only used in plants for seed germination, and also amino acids and proteins are not utilized for energy production in plants. It really depends on the organism to what extent you utilize proteins as energy source. Where proteins are of particular importance, let's say, is when we starve, and especially long-term starvation will actually also break down things like muscle proteins for energy production. The types of starvation proteins are very important, and also when we don't utilize carbohydrates, and when we suffer from diabetes. So this is just an intro to when we really degrade amino acids and proteins for energy production. Okay, so this slide is an overview of how both dietary proteins and cellular proteins are broken down, and what happens really to the components, the functional groups and moieties in these amino acids. So regardless of whether we are dealing with intracellular proteins that are degraded or dietary proteins that we degrade, these polymers are first cleaved to individual amino acids. We'll talk about how that happens later. And then, if you look at the structure of amino acids, we have this alpha amine linked to the alpha carbon, which is removed. So this is common to degradation pretty much of all amino acids, that this nitrogen is removed, and we can think of this removed amino group as ammonia for the time being. Okay, and under physiological conditions, this would be protonated, so that's why we usually represent it as an ammonium ion. So we remove this nitrogen, and this leaves us with structures that here are referred to as carbon skeletons, which are alpha-keto acids. Okay? And these alpha-keto acids can then be channeled into the citric acid cycle, either for energy production or just to provide molecules for, in particular, the synthesis of glucose. Okay, so we will learn later or on Thursday that we can distinguish amino acids based on their fate and what they can be used for, and we can distinguish between glucogenic amino acids, ketogenic amino acids. Glucogenic amino acids can actually be utilized as starting materials for the synthesis of glucose, and I think you appreciate the importance of glucose by now. And ketogenic amino acids lead to the production of acetoacetate and or acetyl-CoA, and can be utilized for the synthesis of lipids and ketone bodies. Okay, so this is the fate, at least in just a few words, of the carbon skeleton. But what happens to the nitrogen that is removed? It can either be incorporated into other nitrogen-containing biomolecules. Okay, so here we're not just talking about amino acids, but also nucleotides and other amine-containing molecules, for example, biological amines. If we have an excess of nitrogen that is produced, in excess of ammonia that is produced, it has to be secreted from our body because ammonia is cytotoxic. Okay? And depending on the nature of the organism we are studying or we are dealing with, either it might be safe to just secrete the ammonia as is into the environment, and this is a case, for example, for bony fish and organisms that live in an aqueous environment, or it has to be incorporated, the nitrogen has to be incorporated into something that can be safely secreted from this organism. And in our case, it's a compound called urea. Okay, so the excess nitrogen that stems from degradation of proteins is incorporated into urea, and urea is then secreted via the kidneys with the urine from our body. As we will see, there's actually a link between the urea cycle and the citric acid cycle, because we have common intermediates actually, and we can call this link between these two cycles the aspartate argininosuccinate shunt, or we can refer to this as the Krebs bicycle, or something called Krebs tricycle. So there's a link, as we will see, between these two cycles. Okay, so first we're going to talk about general aspects of degradation of cellular proteins, then we'll talk about how dietary proteins are broken down, and then we'll take a look at what happens in general to these amino acids to take away this nitrogen, okay, to remove the nitrogen. As I said, we have a constant synthesis and degradation of proteins in our cells, and we refer to this as turnover, and the lifetime of individual proteins can range between just a couple of minutes, hours, up to months. Okay, so there are some proteins, in particular some regulatory enzymes, that have a very short lifetime, let's say in the range of minutes, while a protein like hemoglobin is synthesized by immature red blood cells and will be active in erythrocytes until the erythrocytes age and are removed from the system. And the average lifespan of an erythrocyte is 110 days, so we are talking 3 to 4 months. Okay, and that's how stable a protein like hemoglobin can be. There are, roughly speaking, two mechanisms by which cellular proteins are degraded, namely either non-selectively by lysosomal proteases, so lysosomes pretty much swallow these proteins, and proteases stored in lysosomes then hydrolyze these proteins and produce amino acids. And this is true in particular for those with long half-lives, and we're dealing with particular membrane extracellular proteins here also. Or cellular proteins can be broken down more after they have been marked by attaching a protein called ubiquitin. And ubiquitin is a relatively small protein with 76 amino acid residues. It's highly conserved in eukaryotes and is has pretty much the same sequence in many eukaryotes, which also reflects how important it is. Okay, because if you mess with this protein, then you jeopardize the viability of the organism. So this is just a brief overview of what happens actually when we mark proteins for selective degradation using ubiquitin. And we see that this protein, which is here just shown as UB, is attached to amino acid residues in the protein. It's not quite understood what lets the mechanism or the complex that actually attaches ubiquitin to protein recognize proteins for this tagging. Okay, but fact is that one or typically more ubiquitin proteins are attached to a protein that is destined for degradation, and then the proteins are recognized and bound by a big complex in the cytosol that we can refer to as the proteasome. And the proteasome has two caps here shown in blue, and then a central cylinder where the proteins are chopped up, and overall this whole complex has a molecular weight in the range of about 2 million Borg, which is huge. Okay. So how many ubiquitin residues are attached and how these proteins are really recognized and such is still a hot topic nowadays in biochemistry. There are many groups around the world that work on this. Okay, so this is really enough information for our level here. What I would like to point out is that this so-called ubiquitinylation, difficult word, is actually energy requiring in form that consumes ATP in order to attach these ubiquitin residues. As early as in the 1980s, a group around Warshawski, researcher's name, actually found that there is a relationship between the amino acid residue that you find at the N-terminus of proteins and the protein's lifetime. Okay, and this became known as the so-called N-end rule. Okay. Now this N-end rule has to be taken with a grain of salt, because which amino acid residues actually have a stabilizing effect or may have a stabilizing effect on proteins or have a destabilizing effect depends on the organism, in many cases actually on the organism and the investigation. So what I have here is data from yeast. Okay, if you look at humans, there are some similarities, but there are also differences. I think I have underlined pretty much those amino acids that in a wider range of organisms have either a stabilizing or destabilizing effect. We see that if, for example, we have methionine, glycine, alanine, serine, threonine, or valine being the N-terminal amino acid, then we're typically dealing with rather stable proteins with lifetimes in yeast here greater than 20 hours or half-lives. But if we have arginine or lysine, leucine or phenylalanine, we dramatically reduce the half-life of these proteins and destabilize them to such an extent that their lifetimes are in the range of just a few minutes. So it's not necessarily the fact that we have these amino acids on the N-terminal structure. It must be the fact that this is a signal for the ubiquitination complex to mark these proteins for degradation. Okay. So, let's talk about how dietary proteins are broken down to the level of amino acids. So we take up food, and this food reaches our stomach, where degradation of proteins actually starts. And when food content reaches your stomach, the gastric mucosa actually secretes a peptide hormone called gastrin. It's just 17 amino acids long, so it's a short peptide. And that stimulates other cells in your stomach, namely the chief cells and the parietal cells, to secrete pepsinogen, so the zymogen, the precursor of pepsin, and hydrochloric acid. So your stomach produces hydrochloric acid, produces chloride, ion, and protons. Together these form hydrochloric acid, which acidifies the stomach content, as you know, to such an extent that we can have pH values in the stomach down to pH 1 or so. So between 1 and 2.5 or so is typical or not an uncommon pH value, okay, which is very acidic. The acid actually plays two important roles, namely, first of all, to, let's say, kill organisms that you may have taken up with your diet, with your food, okay? And with this, let's say, reduces the risk for infections, okay, let's say bacterial infections, for example. But the other role of acidifying your stomach content is to denature proteins and to make interior peptide bonds accessible for peptidases and proteases that cleave these bonds. Okay? So, the first enzyme really that is of importance for cleaving such bonds is pepsin, okay, and that's produced from pepsinogen. And pepsin actually cleaves peptide bonds after aromatic amino acids and produces shorter peptides. Okay? And these peptides then reach your small intestine, your duodenum, okay, where your duodenum actually secretes another peptide hormone, secretin, which stimulates the pancreas to secrete bicarbonate to neutralize the stomach content at this point, and let's go to the other side, and the pancreas also secretes quite a variety of proteases, ranging from things like trypsin to chymotrypsin and carboxypeptidases. Elastase are other proteases that are secreted by the pancreas, which all further chop down your peptides. So-called aminopeptidases are actually secreted by the intestinal mucosa. So we have both aminopeptidases and carboxypeptidases, which are exopeptidases, meaning that they remove individual amino acids from the amino terminus and the carboxy terminus respectively. So aminopeptidases chop up peptides starting with the amino terminus, okay, and remove the first one, the second one, and so on. Carboxypeptidases work from the carboxy terminus. I'm not sure to what extent Dr. Baker may have talked about serine proteases, and in this context he may have talked about chymotrypsin and trypsin, but you may remember that chymotrypsin also cleaves after aromatic amino acids, and trypsin cleaves after positively charged amino acids like arginine and lysine. Okay, and all these peptidases and proteases then produce individual amino acids, which in your small intestine are actually taken up by the intestinal mucosa and then are released into the bloodstream and are transported to the liver and also extrahepatic tissues. Okay, so breakdown of proteins, dietary proteins to individual amino acids happens then in the gastrointestinal tract, and then our amino acids are transported throughout the system of bloodstream. Okay, so now let's talk about what happens to such amino acids. And as I indicated before, what we have to do is that we remove the alpha amino group. Okay, that's what happens first to all amino acids. And there are different ways how the alpha amine can be removed. Now all these different mechanisms and enzymes that catalyze such reactions are here summarized or shown under the title amino acid deamination, which is a little bit misleading because a deamination could really be that we remove the alpha amine and produce ammonia, okay? And this is not true for the first item here, or the first type of reaction, which are catalyzed by a class of enzymes known as aminotransferases or transaminases. So these are synonyms. Because what happens here is that the alpha amino group of one amino acid to be degraded is transferred to an appropriate acceptor molecule, which would be an alpha keto acid, to produce another amino acid. We'll talk about this in quite a bit of detail in a moment. But this is not what typically is understood under classical deamination. Okay? We have, however, classical deamination reactions, namely the one that is catalyzed by an enzyme called glutamate dehydrogenase. And glutamate dehydrogenase removes the alpha amine from glutamate and produces alpha ketoglutarate and ammonia. Okay. Now we will see in a moment that alpha ketoglutarate is of crucial importance for amino acid metabolism because alpha ketoglutarate is our primary acceptor of amino groups from other amino acids. So we produce in such transamination reactions a lot of glutamate, and this glutamate can then be deaminated with the help of this enzyme, glutamate dehydrogenase. Another type of enzymes that can also perform deamination reactions is actually found in peroxisomes, okay, and is known as amino acid oxidants, and we will see that they also produce ammonia. Okay, so let's first talk about transamination. That's what pretty much happens to pretty much all amino acids that are being degraded. Okay, so we have an amino acid. Here's a general structure with side chain just shown as R, and we want to remove this alpha amino group, and we utilize transaminase or aminotransferases that recognize this amino acid as a substrate. So with regard to the substrate specificity, we may have differences between the individual members of this class of aminotransferases, but most of these actually only accept one molecule as an acceptor molecule, and it's alpha ketoglutarate. Okay, and it's an alpha keto acid, so we should know the structure now. 5-carbon dicarboxylic acid with an alpha ketone. Okay, 1, 2, 3, 4, 5, and what these enzymes do is they just take, it seems, this amino group and attach it to the alpha carbon and convert this ketone into an alpha amino group. And on the other side, on the other molecule, by removing the alpha amine, produce the corresponding alpha keto acid. Okay? Simple enough, it seems. We'll see that this reaction is a little bit more complicated, requires an important cofactor, which is here just abbreviated PLP, which stands for pyridoxal phosphate. And we'll see the structure and how it is involved in this reaction in a moment. So again, the acceptor molecule that is recognized by most transaminases, alpha ketoglutarate. There are pretty much 2 exceptions to that. There is transaminase that also utilizes oxaloacetate, which we know from the citric acid cycle, amongst other things. As a substrate, and it recognizes as a substrate glutamate. Okay, and if we transfer the amino group from glutamate to oxaloacetate, we convert our 5-carbon structure back to alpha ketoglutarate. Okay, and the oxaloacetate is converted into the corresponding amino acid aspartate. And we will see later when we talk about the production of urea in the so-called urea cycle, that actually one of the two nitrogens in the structure of urea comes from free ammonia that has been released, and the other one will actually be provided by aspartate. Okay, so by producing a lot of glutamate in transamination reactions, utilizing a great variety of different amino acids, we can then produce aspartate, transfer this amino group to aspartate, and aspartate can donate it to an appropriate intermediate in the production of urea. Understood? Okay, so now let's take a look at the mechanism, and overall it occurs in different phases, because what happens in these transamination reactions is that first the amino acid of the amino group of the amino acid to be degraded is transferred to the enzyme, to our cofactor pyridoxal phosphate. So we produce an aminated cofactor in the enzyme and the alpha keto acid. So that's the first phase, and we'll take a look at what happens during this phase. And then the amino group from our cofactor is transferred to the acceptor keto-alpha keto acid to produce the corresponding amino acids. So in most cases it will be glutamate that is produced from alpha ketobutyrate, and a deaminated cofactor in the enzyme. Okay. So we will take a close look at this step, and then I'll just tell you that the mere reversal of these reactions would lead then to the production of glutamate. Okay? So, the cofactor that is required by transaminases is pyridoxal phosphate, which is shown here. The pyridoxal phosphate is a derivative of pyridoxine, which is vitamin B6. So we see that this is based on a pyridine. We see a difference here, a couple of differences. This hydroxyl group is phosphorylated in pyridoxal phosphate, and then also this hydroxyl group here is actually oxidized to an aldehyde. So while this is pyridoxine, this is a pyridoxal, and the Al indicates that we have an aldehyde functionality here. And then it's phosphorylated 5' carbon, so we refer to this compound as pyridoxal 5' phosphate. Okay? And that's the active form of our cofactor. You may remember that I told you a couple of times already that aldehydes and ketones like to react with amines. So what kind of bond is formed when ketones or aldehydes react with amines? Do you remember?
Amide.
No, not an amide. No, not an amine. What did I tell you? You should always say if you don't know the answer.
Schiff base.
Schiff base. Okay? So here we have an example for that. Amines love to react with carbonyls, with aldehydes and ketones, and they produce, often enough spontaneously, typically even spontaneously, Schiff bases. Okay? And this type of chemistry is of extreme importance here for these transamination reactions, because that's what we will utilize actually to transfer the alpha amines first to the cofactor and then to our acceptor alpha keto acid. Okay? And you have to understand this desire of these two functional groups to react with each other. So what happens then if we have such reactions occur is, at least for the time being, to produce this aminated enzyme or cofactor in the enzyme, this structure where we have replaced our aldehyde functionality with an amine. Okay? And we'll see that pretty much the chemistry behind this is the formation of Schiff bases, and then some rearranging and moving of electrons and things like this. And the cofactor in this state we can refer to as pyridoxamine 5' phosphate, because we have this amine structure. Okay, so this is pretty much our starting point. This is our intermediate here, and then we donate this amine from this pyridoxamine again to an appropriate alpha keto acid acceptor to regenerate our aldehyde function. Any questions? Okay. So the next slide is a little bit busy and shows you this first phase where we produce this pyridoxamine intermediate here in our enzyme. And you will pretty much never see this free aldehyde functionality. Okay? So the chemistry that I just described is what is happening, despite the fact that you don't see the free aldehyde here, because the aldehyde is hidden already in a Schiff base that is formed between the cofactor pyridoxal phosphate and an epsilon amine or lysine residue in the active site of the enzyme. Okay, we had situations like that before where, I can't remember what it was, I think hard enough I remember, where we had it was glycolysis, right? Aldolase, okay, where our fructose intermediate is actually bound to an epsilon amine of a lysine in the enzyme to form a Schiff base, okay. Now here we have a similar situation. We have an aldehyde containing component. It's actually the pyridoxal phosphate and is bound covalently to the active site of the enzyme by forming a Schiff base. And it's this Schiff base that is being attacked by the amine coming from the alpha amino acid to form ultimately this aminated form of the cofactor. Okay, so don't be confused if you don't see this aldehyde functionality. It's hidden in the Schiff base that we see here. Okay, so think of this form here of the cofactor as the aldehyde form, the pyridoxal phosphate. Okay, but it's not the free aldehyde here because this cofactor is linked covalently to the enzyme through the formation of a Schiff base with a lysine residue. Okay, but the chemistry is the same. So the amino acid actually attacks this partially positive carbon here, and we form an intermediate where we have a diamine. Okay, here nitrogen coming from our lysine residue in the active side of the enzyme, and here the other nitrogen coming from our amino acid to be deaminated, or transaminated, let's say. And we just rearrange this to form now the Schiff base between the amino acid and where we have regenerated the alpha amino, or the epsilon amino group in the lysine residue in the enzyme. Okay. So if we had a free aldehyde functionality here in the cofactor, namely this, and we would have it react and form a Schiff base with an amino acid to be transaminated, we wouldn't exactly get this structure. Okay, so don't be confused here. Now, what we want to achieve is, remember, we want to transfer the amine from the amino acid to the cofactor. Okay, and if you boil it down to what is happening here, to just simply two things, you'll understand this mechanism much more easily. Namely, we transfer or transform this bond that we now have between the aldehyde form of an amino acid, or Schiff base between the aldehyde form and the amino acid, to an analog where this bond is between the amine form of the cofactor and an alpha keto acid. Okay, and then we just hydrolyze this bond. So if you look at this structure, and I'm not trying to confuse you, I'm trying to make this easier for you, okay? If you look at this structure, you get this type of Schiff base if you have an amino acid attack aldehyde in the cofactor, okay? And here it's called the amino acid pyridoxal phosphate Schiff base, or aldimine, because it's a Schiff base between the aldehyde form of the cofactor and the amino acid. And now, going through this, we actually form first conjugate between these two components where this double bond, the shift base, has been moved. Okay, and it's now can be seen as a shift base formed between the amine form of the cofactor and the keto acid. Okay, so here's the double bond in our aldimine. Here's we move this double bond basically here to this position to form something that is equivalent to what we would get if the amine containing cofactor would attack an alpha keto acid. Okay, and then we just pretty much cleave this bond here between the alpha keto acid and the amine containing cofactor. So how do we achieve this transfer of our electrons? By first simply abstracting a proton here from our alpha carbon in the amino acid component, and this is presumably done with the same lysine residue that previously formed the shift base with the cofactor. Okay, we remove a proton here from the alpha carbon of the amino acid. We form a carb anion, so there should be a charge here. We move these electrons down here, these electrons down here, and go through such a quinoid intermediate. And then we just donate this proton back, but not to the proton that we took it from, the alpha carbon, but our carbon here that is part of our cofactor. You may remember some of the previous enzyme mechanisms that we discussed, that simply taking away a proton here and adding proton here is not an uncommon theme in enzyme mechanisms. Here we see it again to achieve something quite different. So we add the proton to this carbon here and have formed this structure which we can refer to as a ketimine, because it's an imine between our keto acid and our amine containing cofactor. And then pretty much what happens is that we hydrolyze this bond here. It's formulated as a hydroxide ion attacking this alpha carbon. These electrons moving over here and these electrons moving over here, and something can go through this if you want to. We just hydrolyze this bond and form the alpha keto acid and our amine containing cofactor. Okay, so not a big deal really. What we have achieved at this point is to form this aminated enzyme, and from the aminated cofactor, pyridoxamine phosphate, the amine is then transferred in just a reversal of these individual steps in the mechanism to the appropriate alpha keto acid acceptor, which in most cases will be alpha ketobutyrate. And alpha ketobutyrate will then be converted to glutamate. Now, if we have, for example, transfer of the alpha amine from glutamate to oxaloacetate, then oxaloacetate will actually accept the amine from the cofactor and will be converted to aspartate. Okay, but in most cases, for most amino acids, we have a transfer to alpha ketobutyrate and produce glutamate. And the reactions are just a reversal of what we just discussed. Okay, so I said that there are a few exceptions for transaminases that can accept as an alpha keto acid something else than alpha ketobutyrate. I talked about oxaloacetate being converted into aspartate and the importance of that. Another class or another type of transaminase that can accept something else is alanine aminotransferase. Alanine aminotransferase can accept pyruvate as an alpha keto acid acceptor. Okay, and this is of great importance for the so-called glucose-alanine cycle. So, what happens here? We have in extrahepatic tissues, in particular in muscle tissue, glucose as an energy source, which is broken down in glycolysis. We talked about this, and pyruvate is being produced. Okay. Now, this pyruvate can be transported back to the liver okay? And the liver can utilize pyruvate as a starting material for the synthesis of glucose in gluconeogenesis. The pyruvate can be transported in different ways. Okay, it could be transported as pyruvate, but could be converted into lactate, depending on whether or not we produce lactate in the muscle. Or it can be converted in muscle tissue to alanine. Okay, and this conversion actually serves two purposes, namely that it transports basically our pyruvate back to the liver for gluconeogenesis, and it transports ammonia that is produced in the degradation of amino acids in muscles safely to the liver, where this ammonia can be incorporated into urea for secretion via the kidneys with the urine. Okay? So, if we just take a look at this part of the slide, we actually have breakdown of amino acids in muscle tissue. We produce excess nitrogen, let me call it this, and this excess nitrogen is transferred to alpha-ketoglutarate to produce glutamate, okay? And glutamate can then be transaminated again, converted to alpha-ketoglutarate by transferring the alpha-amine from glutamate to pyruvate to produce alanine. And the alanine can then safely be transported. Remember, ammonia is toxic, okay? Alanine can be safely transported in the bloodstream to the liver, where the alpha-amine is transported back or transferred back to an alpha-ketoglutarate to produce for biosynthetic reactions or whatever. But if we have excess nitrogen that we want to get rid of, we produce urea and secrete this via the kidney. The pyruvate then, that is produced in this transamination reaction, can be utilized for gluconeogenesis, for the production of glucose, which can be transported in the bloodstream again to extrahepatic tissues, for example, muscle for energy production. And this is meant by glucose-alanine cycle, very, very important. Okay? Important also because it transports excess nitrogen throughout our system safely. There is another mechanism that allows us to safely transport excess nitrogen throughout our system, and that is to incorporate excess nitrogen into the side chain of glutamate to produce glutamine. And glutamine is also of extreme importance for this purpose, to transport excess nitrogen. Okay, so for that reason, these two amino acids, glutamine and alanine, typically found the highest concentrations in our blood if we look at all amino acids that we have in our blood. And these have much higher concentrations, particularly glutamine. Okay, so this was what happens when transaminases remove the alpha-amine from amino acid and transfer it to alpha-keto acid. Now, what is a true deamination? The first one is shown here. It's the one that is catalyzed by glutamate dehydrogenase. So this occurs in the liver and also in the kidney, in the mitochondria of these tissues. And glutamate dehydrogenase, as the name implies, tells us, is an oxidoreductase that can utilize either NAD plus or NADP plus, can use both, okay, and oxidize here our alpha carbon from amine to imine, alpha imine, so we produce as an intermediate this alpha aminoglutarate, which can be hydrolyzed to remove this nitrogen in the form of ammonia, okay, ammonium ion again, and our corresponding alpha keto acid, alpha ketoglutarate. So here we have then in the liver or also in the kidneys, deaminated glutamate and have produced free ammonia, which can then be utilized for the production of urea. Okay, so as I said before, amino acid oxidases are enzymes that can also catalyze true deamination reactions, and they are found in peroxisomes. for the liver and kidney. And what happens here is that we also have this type of oxidation reaction of our amino acid and hydrolysis to produce the corresponding alpha-keto acid from the amino acid that is being degraded. The cofactor that is utilized by these amino acid oxidases is either flavin or flavin mononucleotide, so the sister compound that only has one nucleotide. This is this AMP part. Okay. What is interesting is that there are amino acid oxidases that can break down or deaminate, I should say, L-amino acids, and others that can deaminate D-amino acids, and they are very stereoselective. Okay, so this is one classic example of stereoselective antibodies. So the L-selective amino acid oxidases will not recognize the corresponding D-amino acid and vice versa. Also, interestingly enough, if I'm not mistaken, okay, the D-specific amino acid oxidases seem to utilize flavin adenine dinucleotide as a cofactor, while the L amino acid specific amino acid oxidases seem to utilize FMN as a cofactor. Okay, but that's just a side note. What happens then is that the reduced cofactors are oxidized to molecular oxygen to produce hydrogen peroxide. Hydrogen peroxide is converted by catalase to safe, non-toxic products, water and oxygen, and we have oxidized back our cofactor, be it FMN or FAD as shown here. So here is the production of glutamine that I indicated before. So again, especially in our body, we have to remember that ammonia is cytotoxic. It's produced in the degradation of proteins and amino acids, and we have to transport it safely to the liver so that we can incorporate it into the safe molecule urea. As I said, alanine is of great importance, and the other molecule is glutamine. Oops, where's glutamine here? So the enzyme that actually utilizes ammonia and incorporates it in the side chain of glutamate to produce the corresponding glutamine, so this daughter compound of glutamate that has the amido group in the side chain, is glutamine synthetase. And we'll see when we talk about biosynthesis of amino acids that this enzyme is actually of great importance for amino acid metabolism. So it's not only restricted to this reaction here, if we look at the big picture, but it's also important in plants and microorganisms for the assimilation of nitrogen, because it can actually utilize ammonia and incorporate it in the side chain of glutamate to produce glutamine and pretty much feed the pool of amino acid of nitrogen containing biomolecules. Okay, and this is of central importance. We do not actually utilize glutamine synthetase for this purpose. We utilize glutamine synthetase to pretty much get rid of excess nitrogen. Okay, and that's shown here. So glutamine synthetase, as the name implies, synthetase consumes energy in the form of ATP, and the ATP is utilized to activate our side chain carboxyl group in glutamate to produce this gamma-glutamyl phosphate. Okay. Now we can incorporate, so we activate this. This is a phosphoanhydride bond. Okay. Not a mixed anhydride, excuse me. We incorporate then the nitrogen here to form an amido group. We have consumed ATP and produced ADP, and the glutamine can then be safely transported from extrahepatic tissues to the liver. And then in liver mitochondria, but to a lesser extent also in your kidneys and also in your intestines, can cleave off with the help of an enzyme called glutaminase, the side chain nitrogen, produce glutamate and ammonia, and this ammonia can be incorporated into urea. Okay? So nitrogen is produced in extrahepatic tissues. We want to transport it. We can incorporate it into glutamine. We transport glutamine from extrahepatic tissues to the liver, and then in the liver we cleave off this side chain nitrogen, we produce glutamate again, and the nitrogen is incorporated into urea. Simple enough. Okay, so glutamine plays also a very important role, and I indicated this previously for biosynthetic reactions, because there are various mechanisms that nature uses to incorporate nitrogen into biomolecules, and one that I mentioned previously, at least briefly, is to utilize glutamine and utilize so-called aminotransferases to transfer the side chain nitrogen from glutamine into appropriate acceptor molecules, and we'll see how that works and what happens in these reactions when we talk about biosynthetic reactions, okay? But this is important, this type of source of nitrogen in these aminotransferases in a great variety of biosynthetic reactions. Excuse me. So this slide again just provides an overview of what we have discussed. So we break down cellular or dietary proteins down to amino acids, and then we remove the alpha amino group to produce the corresponding alpha keto acid. Okay, in this, in turn, in this reaction, we transfer the alpha amine typically to alpha ketoglutarate to produce glutamate. So we can deaminate glutamate with type of glutamate dehydrogenase and produce ammonia. We could also utilize glutamate to incorporate more nitrogen into this molecule and produce glutamine, okay, utilizing glutamine synthetase. We can then use glutaminase to cleave off the side chain nitrogen to produce ammonia. Again, this ammonia will be utilized in our body to produce urea, but I'll talk about this part of the slide in a moment. We talked about the role of the glucosalanine cycle, and we can also incorporate excess nitrogen in extrahepatic tissue, in particular muscle, to pyruvate. Okay, so this is, it's a little misleading, this arrow here, so you may want to add a question mark here or cross this out, so because that's not exactly what's happening. We produce in those tissues alanine from pyruvate, and then can transport alanine to the liver, where we can transport this amine again to alpha ketoglutarate to produce glutamate, and then we can remove this nitrogen utilizing glutamate dehydrogenase. So it's not alanine that is deaminated in the liver. Okay, so this arrow here is misleading. Again, what happens with this nitrogen or this ammonia depends on what organism we are looking at. There are organisms that live in an aqueous environment, and for those organisms it's safe enough to just secrete ammonia into the environment. And here we are talking about aquatic vertebrates, bony fish, and also some large amphibia. So they live in lakes, they live in the ocean, okay? For them, it's safe to just secrete it because the toxic compound is immediately diluted in the environment and doesn't do any harm to these organisms. So they never had to develop mechanisms that are cost intensive. Remember, synthesis of proteins, each enzyme that you need to produce something is cost intensive. Okay? Oops, sorry. Other organisms, so these organisms that produce ammonia and release ammonia are called ammonotelic animals. There are others, including birds and reptiles, that will secrete all excess nitrogen in the form of uric acid. Okay? And what I mean by all excess nitrogen, I'll make clear in a moment. We are ureotelic animals, so we share this with many terrestrial vertebrates and also sharks. We incorporate excess nitrogen that stems from the degradation of proteins in urea, as I pointed out a couple of times before, and secrete urea with our urine. So, if you paid attention, I specified that we secrete excess nitrogen that comes from degradation of proteins in the form of urea. Doesn't mean that we secrete all excess nitrogen that we produce in the form of urea, because we also secrete nitrogen that comes from degradation of nucleic acids and nucleotides. And we actually produce also things like uric acid in the degradation of pyrinucleotides. Okay, we'll see what happens to or nucleotide degradation when we talk about nucleotides in the context of synthesis. Also, they discuss both synthesis and degradation, because, as you know, nucleic acids and nucleotides are not being utilized for energy production, and that's pretty much our focus of our discussion of metabolism now. So we produce uric acid to also get rid of excess nitrogen, but the excess nitrogen that stems from degradation of proteins is, in our case, incorporated into urea. So, I said previously that we are not going to discuss degradation of individual amino acids in any great detail, because it's rather complex. If you're interested in that, feel free to consult your textbook. If you're using the Leininger, even in the Leininger textbook, it's not particularly detailed. If you're using a textbook like Voet and Voet or so, it's a bit more detailed, but it's quite complex, okay? And we don't have the time to do that. What I want to share with you is what happens in general with these amino acids, what's the fate of these amino acids, and how can we classify them. So this slide shows you where the 20 standard amino acids, or what type of degradation products the 20 standard amino acids actually produce that can then be channeled into the citric acid cycle. Okay? And some of these 20 standard amino acids actually produce more than just one intermediate or one of the molecules shown here in this slide. So you may see the names a couple of times actually. Aspartate, for example, you see here, you see it here, and so on. So we see different boxes, so there are a relatively limited number, 7 or so, 1, 2, 3, 4, 5, 6, 7 exactly, different products that can be produced in the degradation of the 20 standard amino acids. Again, some of these produce more than just one of these products, and there is a distinction made by using different colors between certain amino acids. We see that some amino acids here are in these green boxes, and others are actually in these, what is it, salmon colored, ugly color. So the red names here of these amino acids indicate that these are ketogenic amino acids. And the ketogenic amino acids, as I pointed out before, produce acetyl-CoA and acetoacetate respectively. And these compounds, as you know, can be utilized for fatty acid and lipid synthesis and lead to the production of ketone bodies respectively. Okay? That's why we refer to them as ketogenic amino acids. Those amino acids that are shown in these green boxes here produce intermediates that ultimately can lead to the synthesis of glucose, and for that reason are called glucogenic amino acids. Now, if you went through all these different boxes and these names here, you would find that there are actually just 2 amino acids, namely leucine and lysine, that are purely ketogenic. Then there are a couple, namely isoleucine, threonine, tyrosine, phenylalanine, and tryptophan, that are both ketogenic and glucogenic. And all the other amino acids are actually glucogenic. Okay. So I can't remember to what extent I stressed this previously, but there are different ways how you can describe amino acids. I shared with you a couple of terms that can be used to describe amino acids. I defined, for example, standard amino acids / proteinogenic amino acids, which is synonymous. So these are amino acids that are utilized by ribosomes for protein biosynthesis. Remember that? Here you can make a distinction between glucogenic and ketogenic amino acids, which refers to pretty much their fate and the products of their degradation in an organism like a human being. Then you could make a distinction between, for example, natural amino acid and non or unnatural amino acids, which simply describes whether these amino acids are produced naturally, okay, or have to be synthesized in a chemistry lab or something like that. Another distinction that can be made is between essential amino acids and non-essential amino acids, which describes whether or not a particular organism is able to synthesize certain amino acids from scratch, meaning has the enzymatic repertoire to synthesize such an amino acid from scratch or not. If the organism can synthesize a particular amino acid, the amino acid is non-essential because it does not necessarily have to be taken up with the diet. If we're talking about an essential amino acid, we refer to an amino acid that has to be taken up with the diet. And that's something that we will take a closer look at when we talk about the biosynthesis of amino acids. Okay? So don't confuse these different terms because they mean certain things. So I often enough have students that are referring to, for example, standard amino acids, but then they say natural amino acids. It's not the same thing. Okay? Obviously, the standard amino acids are also naturally occurring with this natural amino acid, but try to keep these terms separate and understand what they mean. Okay? Because we will, for example, just in a moment, see some amino acids that we don't find in proteins, but they occur in our body, so they are certainly natural amino acids, but you may never have seen them before. Okay, and we see this right now when we talk about the urea cycle. So this urea cycle primarily happens in the liver, as I pointed out before, and takes place partially in the mitochondrial matrix, which is outlined here by this oval, and partially in the cytosol. Okay. So certain intermediates of this pathway can actually be transported across the inner mitochondrial membrane, which is indicated by these yellowish circles. Now, despite the fact that we refer to this as a cycle, and this is considered a cyclic pathway, there's actually the first reaction here catalyzed by an enzyme called carbamoyl phosphate synthetase. So there are different pronunciations for this name, like carbamoyl or carbamoyl. You hear, so I may switch back and forth if you prefer one, use it. Which is actually not within the cycle, but it's a prerequisite for the other reactions. Okay, and this is first reaction, considered the first reaction of the urea cycle. Okay, but what it does is, and we'll take a look at this in a moment, using a different slide, it incorporates or combines ammonia with bicarbonate. It requires energy, actually 2 ATP equivalents, to produce this molecule that we can call carbamoyl phosphate or carbamoyl phosphate. And this then is utilized within our cycle to produce an amino acid that is not one of the 20 standard amino acids, but it's a naturally occurring amino acid. It's called citrulline. But before we take a look at these individual steps within our cycle, let's first talk about this first step that produces this carbamoyl phosphate. And so this molecule, and it is catalyzed by an enzyme that is called carbamoyl phosphate synthetase 1. So what does this name tell you? I want to hear at least 3 things that you can take away from the name.
Synthetase means you're synthesizing something, but you're using ATP as well.
Yeah, synthetase means it consumes ATP, okay? And we'll see that it actually consumes 2 ATP equivalents. So this is one thing. Tell me something else. simple one. It tells us what the enzyme produces.
There's one standard derivatives and one that's.
Exactly. Okay, so this is called carbamyl phosphate synthetase 1. So we can assume that there's also an enzyme called carbamyl phosphate synthetase 2, and there is, okay? And we will actually talk about carbamyl phosphate synthetase 2 when we talk about pyrimidine synthesis, okay? Because carbamyl phosphate synthetase 2 is actually an enzyme that we require for the synthesis of pyrimidines and pyrimidine nucleotides, okay? Now, we'll see that there are differences between carbamyl phosphate synthetase 1 and carbamyl phosphate synthetase 2, and I will talk about this in more detail when we talk about synthesis of pyrimidines. But remember that there's also a number 2 here, okay? Just as we had 2 phosphofructokinases, right? The third thing would have been really a no-brainer, namely that it produces carbamyl phosphate. Okay, so try to understand why we give certain names to certain enzymes. Okay, here often enough it describes the substrate that we use as product that we produce. Here we have the product, and it's carbamyl phosphate. So what happens in this reaction? This is bicarbonate. Okay, we produce bicarbonate from producing carbon dioxide, and in aqueous environment it forms bicarbonate, and we activate, the enzyme activates actually this bicarbonate by utilizing ATP and phosphorylating this carbonate to form something that we can call carbonyl phosphate. So what kind of bond do we have here? Acyl phosphate or mixed anhydride, okay? Which can easily be attacked here by ammonia, okay? Remember, excuse me, we want to incorporate nitrogen, excess nitrogen into urea. So we attack here and form a carbamate. So this is the structure of a carbamate. And this carbamate is further activated by forming another mixed anhydride here by utilizing yet another ATP, and we form this phosphorylated carbamate, which we can call carbamyl phosphate. And carbamyl phosphate is then utilized by an enzyme called ornithine transcarbamylase. Okay, and this carbamyl, this part of the molecule here, okay, is transferred to the alpha amine, to the side chain amine, excuse me, of a non-standard amino acid called ornithine. If you look at the structure of ornithine, it should remind you of the structure of lysine, because they are only different with respect to the number of carbons we have here in the side chain, the methylene groups in the side chain, because where in lysine we have 4, here in ornithine we have 3. Okay? And this side chain amine here, this delta amine in the side chain, attacks our activated carbamyl phosphate carbon. We cleave our phosphate and we produce this structure here, which is a derivative of ornithine containing this carbamyl group here, basically shown in blue and red, I think it is. Difficult to see from here. That is called citrulline. Okay, so this is a naturally occurring amino acid. It's not a standard amino acid. It's not found typically in protein, but it's an amino acid that occurs in our body. And this citrulline can be transported out of the mitochondrial matrix into the cytosol, where it's combined with aspartate. So you have to look at these structures and formulate these reactions to better understand what's happening here. But we actually have the alpha amine of aspartate attack this partially positive carbon to form this structure, which is called argininosuccinate. Okay. So, first of all, what I would like to point out is that this reaction here that leads to argininosuccinate is catalyzed by an enzyme called argininosuccinate synthetase, and the name again tells us that it consumes ATP. And we need this ATP to actually activate here this position. It's not shown here on the slide. Here our our position here, making it easier for this amine to attack this carbon, and then we form the structure that is to see here is that we consume 2 ATP equivalents in this step, okay? Do I have this here? No, wait a second. Now, why is this molecule called argininosuccinate? Because if you look at the structure, it contains a guanidino group here, and all the other components that make arginine, the amino acid arginine, and it has linked covalently to it a succinate residue, a 4-carbon dicarboxylic acid. Okay? So simply enough, if you know the names and you have an understanding of these amino acid structures and something like succinate, it's not that complicated. Okay? So what happens then is that this bond between our succinate part and the arginine part is cleaved, and what is released actually is fumarate. So it's not succinate, but it's fumarate that is produced of unsaturated trans dicarboxylic acid that we know from the citric acid cycle, remember? And this is catalyzed by a lyase, which we can call argininosuccinate lyase, or we can simply call it argininosuccinate. So this produces arginine. Okay, so what's the big deal? We have produced a standard amino acid, natural amino acid, okay? Yeah, but what we have done is we have incorporated into this molecule here nitrogen coming from ammonia and nitrogen coming from aspartate, which was likely to be produced in a transamination reaction that involved oxaloacetate and glutamate to produce alpha-ketoglutarate and aspartate. So we have removed excess nitrogens, have incorporated it at this point in this structure, which we just hydrolyze here. Okay, so this guanidino group is hydrolyzed here to produce olefin again and produce urea. And urea can then be from the liver transported into the bloodstream, is sequestered by the kidneys, and then secreted with the urine. Okay. So we will see, and that's what I had here, is we will see that this is yet another reaction that we see here in the utilization of aspartate that is, can be used in nature to incorporate nitrogen into biomolecules. So we will actually see this same type of chemistry, or very similar chemistry, when we talk about the synthesis of purine nucleotides, particularly adenylate. Okay. So again, there are a limited number of reactions that can be utilized to incorporate nitrogen into biomolecules. This is one of them. And remember, urea production is cost intensive, and if you take into account these 2 ATP equivalents that we use here in the argininosuccinate synthetase reaction and the 2 ATP equivalents that we use in the carbamoyl phosphate synthetase 1 reaction, it takes us, and it costs us 4 molecules of ATP. Okay. So, real quick, give me 3 more minutes, 2 more minutes, and then we're done with this. So there's this link between the citric acid cycle and the urea cycle. Okay, it's the argininosuccinate shunt, or we can also call it Krebs bicycle or Krebs tricycle. And we link these 2 cycles via this argininosuccinate that can be cleaved to fumarate. Fumarate is an intermediate of the citric acid cycle. It can be converted to malate. Malate can be oxidized to oxaloacetate. So these are all components of the citric acid cycle. Remember that. And oxaloacetate can be converted to aspartate, and the aspartate can then be utilized to produce or to provide more nitrogen for the production of urea. So this is the link, and all these compounds can occur both in the mitochondrial matrix and also in the cytosol. Okay. So just 2 words about the regulation. So there's direct relationship between the concentration of amino acids that you have and the level of amino acids in your bloodstream and the level of activity and the amount of enzymes of the urea cycle. So this is an induction actually that happens here. If you have increased need for the enzymes of the urea cycle because you break down a lot of proteins, you will improve. So there's a linear dependence between the concentration in blood. Makes sense. We break down a lot of amino acids, we produce a lot of ammonia, we have to get rid of that. And this level of induction is typically at the transcription level, and is regulated and affected by hormones like glucagon and glucocorticoids. It's stimulated by these, and it's inhibited by insulin. Now, this is all a regulation of the amount of enzymes that can perform these steps in the urea cycle. The more enzymes we have, the more proteins we can break down. There is also allosteric modulation, so at the individual enzyme level, and this is affecting carbamoyl phosphate synthetase 1, and this is activated by N-acetylglutamate. So if we break down more proteins, we produce more glutamate in all these transamination reactions. If we have increased concentrations of glutamate, glutamate is acetylated with the help of an enzyme called N-acetylglutamate synthase that utilizes the acetyl group from acetyl-CoA to simply acetylate here our nitrogen, our alpha nitrogen, to produce this N-acetylglutamate. And if the concentration of N-acetylglutamate increases as glutamate concentrations go up, this compound here has a great activating effect here on our first enzyme of the urea cycle, the carbamoyl phosphate synthetase 1 reaction. Okay? So, again, as we produce more glutamate, break down more proteins, we have an activation of this first crucial enzyme that leads to the production of glutamate. Okay, so on Thursday, Niati will talk about genetic disorders of the amino acid metabolism, and then I will, if I have time, and I should have time, will talk about start phosphorylation in the active transplant.